Hey you guys, today we're going to be talking about disruptions to the cell cycle and things that can lead to cancer. Now, I know this is a very sensitive topic, and if this is something that hits close to home for you, feel free to reach out to me, or we can talk about things whenever I return. So, your target for today is to be able to predict outcomes of disruptions to the cell cycle by identifying different factors that could lead to the development of things like cancer. So, quickly recapping cell cycle, we started talking about how this is how cells make new cells. We've got two main phases. We got interphase, which is G1, S, G2. Cell is doing normal things. It's going to grow. We're going to double DNA. And we have something called the final checkpoint. If everything looks good to go, then we move into M phase, which is where we go through PMAT. And then we get those two daughter cells at the end of cytokinesis. So, recapping mitosis really super quickly. We have this first phase, which we call prophase. DNA is packed into chromosomes, so we go from chromatin looking like loose spaghetti into these very nice X shapes inside the nucleus. From here, we go into metaphase. This is where we have these spindle fibers, the little lines, and they're going to align the chromosomes in the middle of the cell. Once we've got that done, we jump into anaphase, where now those spindle fibers are going to start pulling those chromosomes apart, moving them to opposite ends of the cell. And then we have telophase, where two nuclei are going to start to form inside of the same cell. Once we've got that, we go through cytokinesis and the cell officially splits. So normally, those cells are going to catch mistakes and prevent errors through that G2 checkpoint that I mentioned. Think of it like going to the dock. So we're checking to make sure, is all of the DNA doubled and copied? Yeah, all right, cool. Did we copy all of the organelles? Great. And did the cell grow to the right size? Perfect. So if everything looks good to go, we continue to mitosis, we go through those steps. However, let's say, for example, maybe the cell didn't grow to the right size. It stayed the same size and it's not big enough. Instead of going through mitosis, this is where apoptosis occurs, where we call it programmed cell death. The cell will purposely explode or burst. It goes through apoptosis. That way, if it was damaged or if there was some type of mistake, it doesn't continue to spread that mistake as it makes new cells. But what about whenever things go wrong? What happens if we accidentally skip one of those checkpoints or we don't catch it in time? So whenever those checkpoints get skipped, those mistakes are not caught. So thinking about this animation from your virtual lab, which side replicated faster? Side B, right? We can see here we're at 16 cells. This one's already at 1,148. So this side is replicating a lot faster. Which one's producing more cells? Again, if we look at it, side B. So side B is making a whole bunch of cells really, really quickly. So whenever we've got this uncontrolled or unregulated cell growth, these normal cells can become cancerous. And when you have a whole bunch of these cancer cells that start just reproducing really, really quickly, it forms this mass of cells that we call a tumor. Luckily, your body has this natural defense against cancer, which are called tumor suppressor genes. So think of these kind of like the brakes of cell division. So normal genes, whenever they've got these suppressors, they're going to step on that brake, they're going to slow down cell division, make sure things aren't happening too rapidly. But other times, with that mutation, that brake doesn't work. So instead of being able to slow down, it just continues to speed up again and again and again. And that's where side B really comes into play, where instead of being able to slow down, they just continue to make more cells faster and faster. So with these tumor suppressor genes, if there's an issue in your body, those genes are going to try to fix those mistakes. They're going to try to stop that mutation, which is a change in the DNA. They're going to go in, and they're going to try to fix it. That way, that cell doesn't become cancerous. And whenever it can't fix those cells, then it triggers apoptosis. So those tumor suppressor genes will tell that cell, hey, something's not right. You need to explode before you spread this to other cells. So that's whenever those cells will go through apoptosis in order to prevent that from happening. But then the question becomes, if we have these genes that protect us against cancer, why does cancer still exist? Our body has a natural defense, but yet cancer is still one of the growing causes of death in the United States. So there's a couple of things that can happen. You can have a mutation to those tumor suppressor genes to make them not work anymore. We've got something called a carcinogen, which can increase your chance of cancer, like things like tobacco, cigarettes, radiation, which end up creating more mutations to those genes. And then there's also a genetic component, and we'll talk about that coming up. One example is like Angelina Jolie, and we'll get to there. And then other examples could be hormone imbalances. So, how does genetics affect cancer? Certain people can have a genetic risk to cancer if their parents have cancer. And like I said, one famous example is Angelina Jolie. She played Maleficent in that Disney movie. And her mom actually had a mutation which is known to cause breast cancer. So, because Angelina's mom had breast cancer, Angelina was at risk of getting that same type of cancer as well. So, to reduce her risk of getting cancer, she had something called a double mastectomy, which is where she had both of her breasts removed before they'd be able to develop into any type of cancerous cells. Other ways that we can get cancer is through those mutations that I talked about earlier to those tumor suppressor genes. So let's say normally you've got those genes and you've got two of them and they do their normal function, but through the exposure to carcinogens, which are those things that cause cancer and mutations, so UV radiation, smoking, too much exposure to sunlight, any of these things can cause mutations to those genes, which means now they don't work anymore. And when those genes stop working, they don't stop the cancer from spreading. So now those cancer cells can really start to spread all over the place. Sometimes we've got other disruptions to the cell cycle, which might lead to other diseases aside from cancer. So for example, if we've got a disruption to G1, maybe for example, the daughter cell is missing some organelles or it didn't grow. So in this case, instead of copying both mitochondria, we only have one. In the case with S phase, it could be that now the cell's got a mutation or it's missing some DNA, which would be something that would destroy the function of those tumor suppressor genes. Over here, it could be the cell is abnormal in size, right? So maybe it didn't grow to the proper size. Or then in M phase, maybe those cells didn't divide properly. So these disruptions to the cell cycle can lead to other genetic and autoimmune diseases like Alzheimer's disease. We've got abnormal cells. Anemia, you've got fewer red blood cells. They're a lot smaller because they didn't split properly. They didn't grow to the right size. And then stuff like Parkinson's, which is an autoimmune neurodegenerative disorder where you've got tremors, you've got slowed movements, a whole bunch of just really bad things start to happen whenever we've disrupted that cell cycle. I have one more question for you in this video, and that is, can cells live forever? I'm going to have you read about this woman named Henrietta Lacks. She was born August 1st, 1920. She died October 4th, 1951, so almost exactly 73 years ago today. Or did she? I'm going to have you read about Henrietta Lacks below to find out, is she actually still alive today? All right, good luck.